We move to the as third presenter today. It's Professor Roni Aloni with the School of Plant Sciences and Food Security in Tel Aviv University, Israel. The title of his presentation is Enhancing Tree Growth and Wood Production with Plant Hormones. Professor Aloni, the Zoom floor is yours.
Thank you very much. I want to thank the organizers for inviting me to give the talk, and I am pleased to see friends and colleagues, and I'm going to the lecture. And actually, trees grow and wood is produced by growth hormones, by hormones, by signals that are produced in specific cells that we shall see them very quickly. I want to let you all know that I have just published, or Springer have just published a book of 20 chapters that I have written about vascular differentiation and plant hormones. And in this book, you are going to find much more than I'm talking, and I suggest to use it for research and for teaching. Now I would like to start with these two photos, and I'm going to show you to show them in a higher magnification. The primary hormone or the primary signal that induce plant growth and wood formation is auxin. Auxin is produced in young leaves, and in this photo, I was lucky to see how auxin is synthesized in a lobe. Auxin moves in the beginning by diffusion, becomes polar, and induces a vascular bundle. And this is the main signal from the young leaves. When we go to the lower parts of the plant, here we have the tips of a root, and this is the root cap. And in the root cap, there is a production of another very important signal. This signal is cytokinin. Cytokinin that is produced in the tip moves upward in the vascular system. It is concentrated in the cambium, promotes cell divisions, and the cambium produces more cells, and from these cells, other signals induce the vascular tissue, the wood, and the phloem. When we speak about the movement of cytokinin, the cytokinin which is produced at the tip goes up in the vascular tissue. If I covered a plant with plastic, there was almost no transpiration. Cytokinin didn't arrive to the young leaves and flowers. And if before the analysis, the plants were, the plastic was removed and very gentle wind was blown, cytokinin went up. It's only to tell you that when you have trees in the forest and when they move by the wind, the leaves and the upper parts of the plant get the cytokinin from the roots. The third very important signal that induces wood is Giberline. Giberline is produced in maturing leaves. The mature leaves produce the Giberline, which moves in a non-polar fashion. It moves into the stem, it moves up and down, and promotes cambium activity and induces, specifically, it induces fibers and also in conifers, it induces tracheids. Here I show you the production of short fibers from parenchyma cells, and this is after 10 days. After 60 days, you can see how the fibers elongate, and all this elongation occurs by the influence, is influenced by Giberline. A simple experiment can show you, I don't see here too much, can show you the effects of auxin and Giberline on fiber differentiation. On the left side, you get all these plants, the upper parts were removed, all the leaves were removed, only in this, on the F treatment, I left the leaves, and you can see that below the leaves, there are numbers. These numbers are fibers. When I didn't apply hormones, I only applied the lanolin. In the lanolin, I applied the hormones. There is no any effect on fibers. Auxin by itself almost don't show any clear effect. Giberline by itself also don't show any effects. The combination of auxin and Giberline in low magnification, in low concentration, induce the whole amount of fibers like the leaves. Only to show you that although Giberline is the specific signal for fibers, it needs a background of auxin. Here I'm showing you an experiment on Canal plants, and in this experiment you see plants that are 4 months old. They have many branches, and they show the normal height of these plants under the experimental condition. When I applied a combination of high Giberline and low auxin, you can see this is exactly the same experiment. The plants were higher. They didn't produce any side branch because of the auxin, and the Giberline promote growth. And I'm going to show you the effect on the wood. And when we see the effect on the wood, this is the control, the intact plants, and these are plants that were treated with Giberline and auxin. You can see much more xylem is produced, and this xylem has a different structure of lignin. To say that that we can apply the hormones by spraying, or we don't have to spray, we can we can add the Giberline to the soil. The leaves produce enough auxin, so the Giberline that is non-polar will go up and promote wood formation and fiber differentiation. Here it's only to emphasize with another experiment the non-polar effect of Giberline. And in this experiment, you can see the plant with no leaves. The anatomy or the cross sections were taken where you see the blue bar, and I left 2 internodes above the internode that I analyzed in order to produce some auxin. And with no Giberline, you can see a few vessels that differentiated, but no fibers. When Giberline was added above, you can see fiber differentiation in the xylem, in the wood, also inside, because these plants Also, this is Nicotiana tabacum, as also inside fibers and also fibers in the phloem. When the gibberellin was added from below, the same results, a lot of fibers in the xylem, only to say that mature leaves that can be in the lower part of the crown, that when they produce the gibberellin will move down and up and produce fibers and wood all along the plant. I'm not going to give you too much information because you are not working on molecular biology, but here I'm showing you the process or the biosynthesis of gibberellin. And in the later stages, you can see that an enzyme, a G20 oxidase, G20 oxidase is the rate limiting enzyme. We can manipulate the plants and increase the level of GA20 oxidase. On the other hand, once we do it, we have a lot of gibberellin in the plant, and then another enzyme starts to work, and this is GA2 oxidase, which inactivates the gibberellin. So what you are going to see an experiment when we overexpress the GA20 oxidase and also the GA2 oxidase, that is the deactivating enzyme, and you will see the result. This is an example of our work enhancing plant growth and fiber production by silencing GA2 oxidase. And what you can see here, these are the results. You can see in this direction the wild type. This is the GA silenced, the GA overexpressed, and the GA2 and the GA2. This is a cross of these two lines. So we have GA2 that is silencing it stops the deactivation, and GA20 that promote gibberellin production. And if we see in this direction, it's the upper, this is the high internode, middle of the plant, and this is the lower part. And when we analyze the wild type compared to the silenced one, we don't see any effect. When we increase GA production, gibberellin production with the GA2 overexpressed, we double the amount of wood. When we add the GA2 that doesn't allow the gibberellin will decrease, then we get lignified fibers, and the wood is much stronger. If we go to the higher internodes, for example, when we increase the level of gibberellin in a young internode, we start to get fibers. But when we don't allow gibberellin to be deactivated, we get a stronger effect. I want now to go to another system, and in this system to go to a conifer. This is a young pine seedling. What I'm doing here, I made a cut and separate half of the hypocotyl. In this place, in this point, I add hormones and analyze the differentiation of tracheids from parenchyma cells in this area. And in this photo, you can see a short tracheids that was induced from parenchyma cells with the lignified secondary wall thickenings. in a low magnification, you see that the auxin by itself induce many short tracheids from parenchyma cells in the pine. However, when we add Giberline with a background of low auxin, we start to get a elongation of these tracheids. You can see how much they elongate, and we get a kind of mature wood. So we here have a kind of juvenile wood with only auxin, and when we add Giberline, the effect of mature leaves, we get a longer fiber, a longer tracheids, like in a mature wood. To summarize, what we have just seen is the normal evolution of tracheary elements. In primitive trees, in conifers, we have the tracheids that are induced by auxin and gibberellic acid. During evolution, cells become more specialized, and we get from only auxin, we start to get vessels. Vessels are induced by auxin by itself, the auxin that is produced in young leaves. On the other hand, Giberline became the specific signal for fibers, but it needs some background of auxin, so Giberline is the background, is the specific signal of fibers. And what we can see from this, from what I've shown you, that first young leaves produce one signal, that this signal is the specific signals that produce the vessels. Mature leaves produce another signal, which is the Giberline, that produces the fibers. And in conifers, we have the combination of both signals, auxin and Giberline, that induces the tracheids. And we can apply the hormones by spraying or by watering the plants with Giberline. And today we can also manipulate the plants that we want in such a way that we will get more tracheids or trees with a higher level of fibers. And by understanding the mechanisms, we can manipulate plants, and we can get better plants, either industrial plants mainly for fibers, or improve tree growth and get the type of wood that we are interested in. So I want to thank you and also remind you again that much more information you will find in my 20-chapter new book that gives you much more information than I was able to give you in about 15 minutes. Thank you very much.
Thank you, Professor Aloni. We have time for one question. If anybody has any questions for Professor Aloni, or you can, if you have something later, you can actually get, communicate with him directly through email or Zoom.
One question.
Oh, okay, go ahead.
Good talk. I think 10 years ago or 15 years ago, I read one paper, and I tried to do similar work. Is try to detect pressure inside the cell, how that cell influence their growth. So we know is the environment will be control how plant to grow. So one research, they showed that they can measure exactly pressure inside the individual cell. Do you learn anything about that? How can you comment that type of pressure?
At the time that the plants differentiate, for example, if a vessel differentiates, there is an increase in the pressure in the vessel, and if this vessel differentiates slowly, it will increase in size for a very long time. And if the cell gets higher levels of auxin, it will differentiate very fast, and a secondary wall will differentiate rapidly, and the cell will not be able to increase in size. So if you go along the plant from the young leaves to the root, what you find that because you go away from the young leaves, you go away from the auxin, the level of auxin in the lower part of the tree are reduced, the rate of cell differentiation, the rate of vessel differentiation is decreased. There is more time that this pressure will increase the vessels, and the results are wide vessels in the lower part of the plant, middle size vessel in the intermediate places, and relatively narrow vessels in the near the young leaves.
Thanks.
You're
Great,
welcome.
thank you.